Okay, it's now 2 minutes past 3, so let's get started. You are very welcome to this webinar hosted by ClueCore and Clarivate, where we will investigate the mechanism of Aristolochic acid 1 induced nephrotoxicity. And I'm Ann Holmberg from ClueCore.
And I'm Peter Rebel from Clarivate.
The agenda for today is that we will start with an introduction, giving you a brief overview of the two softwares, and then an introduction to the biological background of today's topic. Then we will do a live demo of the analytical workflow using both our softwares, and we will end with a Q&A session. And for that, please use the chat that you will find. So ask your questions in the chat. We will have a look at that at the end of this session. And the aims for today is to show you a workflow on how to use Qlucore Omics Explorer and MetaCore in combination to first identify genes that indicate a toxic process and then explore the molecular mechanisms of AA1-induced nephrotoxicity. And the first part of the workflow, we will use Qlucore Omics Explorer. And with Qlucore Omics Explorer, you can inspect, visualize, and explore your large biological data sets. You can look for outliers, you can identify structures in the data, and also generate new hypotheses if you find new interesting signals. And once you have a hypothesis, you can perform statistical analysis and, well, report your results. So you can find discriminating genes, metabolites, proteins, using statistical tests such as t-test, ANOVA, regression analysis, and more. Then you can visualize your results in a number of different plot types, and then you can export your plots and results as publication-ready material. You can also perform pathway analysis using the built-in gene set enrichment analysis, as well as to build classifiers using the built-in machine learning algorithms and then use these classifiers for prediction. So in short, Qlucore Omics Explorer is a very fast and easy to use tool to analyze and explore data without being a statistical expert. And then Peter will present MetaCore.
Okay, so MetaCore is a tool that helps you understand what's going on behind those results. So you might complete your analysis in ClueCore, upload the data to MetaCore, and here it's all about exploring the signaling pathways that explain results in omics data sets. So as you can see over here, you can interact with MetaCore by searching and browsing using keyword searches as for any database, but also you can upload an omics data set, and that's what we'll focus on in this demo. Once you upload the data set, you can analyze that data set using several different workflows that are built into this tool and allow you to understand interactions between molecules, signaling pathways that are provide the context for which diseases or toxic processes or normal biological processes are taking place in your data set. And all of that analysis is built on at the back end on this large data set, now over 3 million manually curated interactions. So this is where Clarivate scientists read the scientific literature and where they find experimental evidence for an interaction between molecules or between a disease and a gene and so on, that these interactions are created as this network database. Okay, so in summary, MetaCore, useful to search and browse and get an understanding of a disease process, but also to upload your omics data to analyze and understand the biology that's driving those results there. So in the next slide, we move on to an introduction to the scientific topic. So the reason we chose this topic was because it's followed on from a publication from a group in King's College London that used ClueCore Omics Explorer in combination with MetaCore to explore the biological process behind Aristolochic acid-induced toxicity. And so because they'd use it, we thought that was a really nice demonstration of how these tools work together. And so we'll be guiding you through the step-by-step process of analyzing a data set using these two tools. Okay, to the science. Over here on the left-hand side, we have Aristolochia, which to me is shouting out, "Do not eat me." But nevertheless, there is a compound in there, aristolochic acid, which is used in traditional medicines and is also toxic at the same time. The toxic effect is cumulative, it's chronic, and it remains with you through lifetime. And you can see here over on the right-hand side this illustration showing that the Aristolochia induces nephropathy, which can lead to renal failure. And it also induces renal cell carcinoma and urothelial cancer. Okay, so for the cancer, there's a reasonable amount known. The aristolochic acid, a metabolite of that, binds to DNA and it drives mutations in the DNA. And in particular for this cancer, there is a specific gene signature there's always mutations in the TP53 gene, which encodes the tumor suppressor. So you can imagine that if you were to take a herbal medicine or natural remedy that contains this compound then binds to your DNA, drives mutations in the tumor suppressor gene, then these particular cells that are exposed to this compound become more prone to cancers. For the nephrotoxicity, much less is known. But as shown in the next slide, there is some evidence that P53 might also play a role here. So what is known is that aristolochic acid causes toxicity in the nephrons, and in particular, it damages the proximal region of the nephron. And so you can see in this diagram, we have the glomerulus, which is all about absorbing waste material and solutes out of the blood, transferring it into the nephron. And then as the waste and solutes pass down the nephron, then useful material such as ions, sodium ions, and glucose, amino acids, and low molecular weight proteins are reabsorbed back into the blood, so they're not wasted, and the rest passes through into the urine. And this inset shows a proximal tubule epithelial cell with a brush border designed to improve the reabsorption of useful solutes back into the cell to be recycled into the body. But it also shows how toxins can affect these cells. So a drug or a compound such as aristolochic acid comes in from the bloodstream in through the basolateral side of these epithelial cells and is taken up into the cells, where they can affect the lysosomal function. They can affect the absorption of the useful solutes. They can affect mitochondrial function, redox balance, and so on. And this will then lead to stress and damage in the cells, and then the cell fate decision of whether to repair or go into apoptosis to kill the cell. So that's the drug-induced toxicity. Now, these researchers in the King's College London, when they asked this question of what is the mechanism of this nephrotoxicity, they injected mice for 6 days, once a day, with either water as a control or aristolochic acid. And after the 6th day, they harvested the kidneys and did microscopy of the proximal region of the tubule of the nephron. So what you're seeing in this histogram is on the y-axis is a semi-quantitative score of the extent of damage in these cells. So straight away you can see no damage in the water controls versus damage when aristolochic acid is injected into these mice. So this confirmed what is known, which is that this compound is damaging these cells, and that's the mechanism for the nephrotoxicity. But interesting to hear as well, you can see the difference between the wild type TP53, as it's called in mice, the wild type gene, versus the heterozygote and the homozygote. So the homozygote has the double mutation in both copies of the gene. And you can see this gene dose effect here with more damage occurring when P53 is knocked out, therefore suggesting that P53 plays some protective role. Not completely, you can see in the wild type that there is a considerable damage going on, but P53 provides some additional protection. And so what this team did then was they did omics experiment, transcript omics experiment to look at differential gene expression for water control versus aristolochic acid-induced toxicity, and then comparing the different mutation statuses of the TP53 gene in these mice. And so this shows the data set that was used. It gives details of the data set, and it's a publicly available data set on the gene expression omnibus. And in the next slide, we'll move on to the demo to show how these tools are used for this analysis. Back to you, Anne.
Thank you, Peter. Yes, so we will start by using Qlucore Omics Explorer now to analyze, sorry, the menu row falls down here, to analyze the gene expression changes in response to the exposure to Aristolochic acid 1. So first we will compare kidney tissue that has been exposed to AA1 and versus kidney tissue that has just been exposed to water, and see if we can find any differences there. And then we will analyze the role of P53 by focusing on the toxin exposed genotypes to see if we can find anything there. And then we will export our genes of interest in lists in MetaCore ready format for upload to MetaCore, and then we will move over to MetaCore and Peter will explore the underlying biology here. So let's move to the software. Here is Qlucore Omics Explorer, and I will load the data set. I have it here. So it has been pre-processed a bit into one file that is easily loadable. And the background, as you saw, were Clarium microarrays. Okay, and here now the data set loaded, and what we can see is 29 samples. So each blue dot here is a sample, and we see the samples in a PCA plot, and we have 29,000 gene expressions here that decide the position of these samples. And we can see that the samples divide up in two groups, and the plot that we are using here is a PCA plot, principal component analysis plot. And in a PCA plot, you look at your data in 3 dimensions, even though you have many, many more dimensions in your data set, and you focus on finding clusters. For samples that are close together, they are similar, and samples that are far apart, they are different. So it's a very easy principle. So we see that we have 2 very clear groups here. So let's try to find out what these groups are. So I have one piece of information here that tells us what these samples have been exposed to. So let's color on that. And then we see that all of these samples have been exposed to water, and all of these samples have been exposed to the toxin. So already here we can see that there is a big difference in gene expression between control and toxin here. And if we would like to explore that and find the significant genes, the discriminating genes here, we can do a t-test on exposure pick one of the groups, and then just drag the statistical slider here to set a good statistical cutoff. So p-value and q-value. The q-value is the false discovery rate or the corrected p-value, and that's really what we are focusing on. So with a q-value of about 0.05, we find 8,000 significant genes here. I want a shorter list, so I just continue to filter down to a list which is somewhere between 1,000 and 2,000 genes. And then we see that the separation becomes better, and then we open up a new synchronized plot and make that a heat map where we can actually see the expression of our individual genes. So I will hierarchically cluster the heat map and also color it according to this. So then we see here also that we get two very clear clusters. So that really supports what we see here. And we can see that we have up and down regulated genes in both groups. And if we zoom in a little bit, we will actually see the names of the genes. So let's change that to gene symbol like that. And we can inspect here and see what we see. That. If I would like to see my data in a volcano plot, I just open up another synchronized plot and make that a volcano plot like that. Here we are. And here we can label our genes of interest. For instance, I am interested in what these are and what these are. And let's change the label again to gene symbol. There we are. And I can easily create lists here by just circling my genes with the mouse. I can also add, so let's say here 12e minus, sorry, 10e minus 12 or something like that if I want to introduce another limit here. And this is also interactive. So if I change the cutoff now for the fold change, you see that the plot changes while I'm filtering here like that. And if I maybe would like to have a look at my best genes here, I have them here in my list. So let's add the gene symbol and let's add the Q value and maybe also the fold change to my list. Let's sort on Q value. So here I have a lot of information and I can easily export this list. So these are my best genes. So let's open up a yet another synchronized plot and make that a box plot. So we select box. And then we need to configure the axis. I would like to have exposure on the x-axis, and then I will pick the genes that I would like to show. So here I pick maybe my 4 best, and they are shown here in box plot. Again, I want to select gene symbol as the label here. I might want to show the individual data points, maybe not show the colors and so on to configure the plots. So you see that you can visualize your data in a number of different ways here during your analysis. So these were just a few examples. So let's move over here now, like that. Now we will have a closer look at the different genotypes. So there are 3 different genotypes, as you heard, 3 in each with each exposure. And now maybe I would like to focus on only one genotype at a time. So I will start with focusing on the wild type. So I deactivate all the other ones. So right now I'm looking at the wild type treated with toxin and wild type exposed to water. And then I will do a t-test on that. So wild type compared to all other groups, in this case water. And then again, I drag the statistical slider to a cutoff that I would like. I would like a cutoff of 0.05. I can type that in. And I would like a fold change of 2. And that leaves me with 1230 genes. And if we have a look at them in the heat map as well, we have them here. And you see it's a combination of up and down regulated genes for each of the groups, but a very, very clear separation. Let's color here. So these are the two groups now. And now I would like to export this result to MetaCore. So I right click in one of the plots. I do copy to clipboard, export MetaCore symbols. So it's copied to the clipboard, and then I just go into Excel, for instance, paste this, and then, so here I have the gene ID, and here I have the log2 fold change. Then I do a save as. So wild type, a 1 versus water, like that. And I want to save this as a tab separated txt. So tab delimited txt, like that. Save. And then I can do the same for the other groups. So if I want to do this for the heterozygotes, I just select them, and then I do the same exercise here. So heterozygote exposed to the toxin, and I set the cutoff to the same 0.05, and then I set the fold change to 2, and here we have the results. And then I right click and do the same thing here, export MetaCore symbols, and create a list for that. And then I can do the same for the homozygotes as well. So in this way, I have created 3 lists, and which I have sent to Peter that he will use in his demo shortly. Before we go there, we will explore the role of P53 in the exposed genotypes. So let's do that. So I reactivated everything here, and then we will remove all the water samples. So I just do that by unticking all the water samples, and that leaves only the toxin exposed samples from the three genotypes: heterozygote, homozygote, and wild type. And now if I do an F-test here, multigroup comparison, I want to see if I can find genes that are good at separating these groups from each other. And I just drag the statistical slider again. And now we see that with a cutoff of 0.05, I find 300 genes. And let's have a look at the heat map as well. Let's maximize it a bit here, like that. Maybe change the label here to genotype. So what we see here now is that already in the PCA plot, we can see that the homozygous samples, they are far away, and these are quite close to each other. That's what we see in the heat map as well. So the gene expression of the homozygotes here is very different from the gene expression in the other two groups. So that really supports what Peter showed in the previous slides, the finding from King's College that homozygotes have a different effect than the two other groups, so maybe indicating that we have the protection of p53 here and less or no protection here. So that's another thing that you could easily explore here. And by that, I hand over to Peter to show how you can use MetaCore to now explore the underlying biology using these gene lists that I just created. So over to you, Peter.
Okay, somehow I don't have the same privileges as
Okay,
earlier.
so I will just try to make you the presenter. So now you are the presenter.
Okay, great. Now I can. Okay. So now I'm going to switch over to MetaCore, which is the tool we use to analyze the differentially expressed genes that Anne showed earlier when she separated them and passed over those folders that were MetaCore ready format. So in order to start, then I would come through to this upload workflow here. Over on the right-hand side, we have a toolbox, and over on the left-hand side, we have a space where I can store my data and activate it for analysis. So when I have completed the analysis in QlueCore Omics Explorer, I would come to this workflow here where I upload experiments with my gene ID. And this takes me through a step-by-step workflow to upload the data. And you can see the types of identifiers that are accepted, and including in there are those gene expression tags that we saw in the Clarion microarray chip. So I would go through, browse for the file, select it, then specify that this was an experiment done in mice, so it would map to the mice genes, and then this workflow will then map the data through to the genes which are objects in MetaCore. Okay, I won't go through that full process now, but what I have done is I've already uploaded and saved it to a folder which I've labeled here for Aristolochic acid 1 liver toxicity. And these are now activated using the activate button here. So they're ready for analysis. And I'm going to analyze all three at once. So just as a reminder, we have Aristolochic acid versus water differential gene expression with a data set for wild type, for the heterozygote, and for the homozygote in p53. Okay, the workflow I'm going to use today is the toxicity analysis workflow, which is set up to analyze and provide a report on toxic events taking place in this data set. So the first step of this workflow is just to validate that we've got the correct data activated. And then the next step allows me to filter the data set. So I could filter by tissue expression, disease, and so on. I could leave it unfiltered because we already have this data extracted from specifically from the kidney of these mice. Then this next step allows me to specify any genes that I want to highlight in the data, in the report. So I don't know if p53 is actually in my results, but I do know that p53 is part of the experiment. And so I'm going to make sure that p53 is highlighted in the results set. So here I search by the gene name, and I select p53 itself and the mitochondrial p53. And you'll see how that turns out in the analysis itself. And then as a final step here, we've got, we select which toxic process. And then this will take me through to this page where everything is set up and ready for the analysis. So we have, you can see here, these experimental data sets. You see the numbers of objects which correspond to the genes. These are the proteins, if you like, in this case. And these were the differentially expressed ones from each data set. Over here, I could provide some settings, but they've already been done in QlueCore Omics Explorer, so there's no need for me to touch any of this. And now I apply and to start the workflow. This workflow can take a little while to calculate. So I've actually already set this up here in terms of and completed the calculation here. So what we're looking now is a report comparing these three data sets and providing information on these processes, these toxic processes and biological processes here. So first thing to look at here is the comparison. And what this is telling me is that we have these three experiments here, and that there are 762 genes that are either over or underexpressed and in common for all of these data sets. So these are genes that represent a response to the toxin irrespective of p53 status. So they're agnostic of p53. So these would explain that initial jump in that histogram of what is driving aristolochic acid toxicity. Then in the middle, we have genes that are common to two, but not all three data sets. And then over on the left-hand side here, we have ones that are over or underexpressed and unique to each different genotype status here. So this is the wild type, the heterozygote, and the homozygote. So that says that there are 197 in this comparison that are unique to p53 and response to aristolochic acid. So I'm going to show you two samples of, or two types of results from this workflow. First, I'm going to focus on what is common that provides some of the common toxic pathologies for the for aristolochic acid exposure, and then I will focus on some of the interactions that are specific to p53 wild type and not in others. Okay, so what I've done here already is just by doing a right click, you can see this functionality here. It allows me to distribute by this set. So I've sorted already this subset of genes here to float those to the top of the analysis. So I'm going to just show you two components of this report. One, I'll talk about the toxic pathology biomarkers, and the other one I'll talk about is networks. So here in this biomarkers analysis, we have these are pathological processes that have been created by our team of toxicologists. And then as they analyze the scientific literature and identify biomarkers of these processes, our team annotate these processes with the biomarkers. So you can see, for example, kidney pelvis dilatation. Over here in red, there are 24 biomarkers associated with this process, of which 17 belong to all three over here process toxic reactions. So this is highly enriched for this pathologic process for kidney pelvis dilatation. Now, kidney pelvis dilatation is a structural change in the kidney, which is a general marker of stress response in the kidney. And you can see as well as we come down these really significant enrichments here of these more specific processes. So we can see that in common there is mineralization taking place. So for example, calcium deposition in the kidney, uric acid deposition in the kidney would lead to this type of toxic process. What we can see in green is that this toxic process here, we have cell regeneration, and it's marked in green because p53 is also a marker of that process. So we have highly enriched genes that are common for all three. So it's telling us that cell regeneration, in particular nephron regeneration, is an important process in exposure to aristolochic acid. And at least two of these enriched processes do use p53 as part of that. Okay, so that gives you some guidance of the ways of interpreting the types of information you can get from this part of this analysis, this report. There are many other reports in here. There's toxic pathologies by biomarkers or by drug metabolism enzymes or by process. There are pathway maps, that provide signaling pathway explanations, and I'll come back to that in a minute. There are gene ontologies, so this is using the gene ontology enrichment by gene ontology process. And you can see, for example, here we have small molecule metabolism, organic acid metabolic processes, and all of this makes sense in terms of the Aristolochic acid, organic acid, toxin, and the induction of processes meant to try and clear the body of this toxin, and so on as we come down. Now, I want to come through to networks because I think this is a really neat analysis here. So what we're looking here with a network, what I've done is I've selected one of the networks that represents the genes that are common for all three genotypes. So this is irrespective of p53 status. And what you can see, this network is built off the fly using those 3 million interactions that are in the database and using those 762 genes as a seed. And it's built this set of highly significant interactions here. And you can see straight away from this figure that we have this key driver of all of these genes here. So what you see here is blue with blue indicators, and with a red indicator in this case, is over and under expression of all of these genes that are represented in our data set, and all of them being driven by HNF4 alpha transcription factor. So immediately from the analysis, we're beginning to draw some biological inference. So we know now that HNF4 is likely to be a key regulator of the response to aristolochic acid, and that this is likely driving the response that is agnostic of p53 status, because I selected that set of genes for this analysis. So it's beginning to give me a hypothesis to go away and explore further as to what potential role we have for this transcription factor in driving the response to the acid, to the toxin. Okay. Now I want to come to a slightly different perspective on this workflow. So what I've done here is we've come back to the analysis that was done in this toxic analysis workflow. And this time I switched the perspective. So where up until now I've explored the 762 genes that are in common, you'll see below here that MetaCore has also done a direct comparison of the wild type versus the homozygote, because it found some significant differences between these two. And that validates what Anne had shown earlier, in that you tend to get the heterozygote and the wild type clustering together and a greater separation from the homozygote. So I'm interested in this analysis here. So I've selected this subset here, this analysis. And then what I've done is I've said, right, float to the top all of the interactions that are most enriched for this subset of genes that are specific to p53. So what this will tell me is what are the pathways and the processes that are operating in wild type mice that are not operating in the homozygote mutant. So if we come down here, we can see pathways and processes that are operating here. We can see lipid accumulation. So that's a toxic process that's specific when p53 is present, but not when it's not present. But importantly here, you can see we've got inflammatory response, we've got glomerular injury, we've got tubular inflammation, so we've got inflammatory response and damage. All of these in green because they also p53 dependent responses. So really significant result here, saying these p53 dependent responses are coming to the top of the analysis. And what's more, that coincides with the fact that we know that this is a p53 specific set of indicator genes. Okay, I'm going to come down to the pathway just to wrap up, because I promised I'd show you a signaling pathway map. And this again tells us a very nice story, because you can see there are two pathway maps that are standing out at the top with the most significant enrichment here. These G protein and GTP metabolism, implying that G protein signaling, GTP metabolism and activation of this pathway are really important in regulating a p53 dependent response to the acid. And look at this. The third one here actually has p53 in the title, and it's highlighted in green. So this is the one I'm going to explore and show next. Okay, so here's the pathway. What it's doing is it's loading the experiment on top, so you can see in highlight in bold those genes that coincide with our experimental data set. P53 is highlighted because when I set up this workflow, I said, even if P53 is not in my data set, highlight it for me, please, and that's what it's done. And look at this, MDM2, which is overexpressed in the wild type, overexpressed in the heterozygote, and not at all expressed in the homozygote. So this is telling me straight away that we have a gene from our experimental data set that is interacting with P53. We do a mouse over the interaction and it gives us more information. It's telling us that MDM2 binds to P53 and uses the ubiquitinylation and degradation of this gene. So it's giving me another mechanistic hypothesis to go and explore, that aristolochic acid is inducing toxic reaction in these epithelial cells. That toxic reaction is probably dependent somehow on MDM2, which in turn drives a P53 response, and P53 in turn regulates that cell fate of repair or apoptosis, and that's defining that response in terms of damage or reduced damage. So we're beginning to build that mechanistic story around the acid response, the microscopy work, the differential gene expression, and now molecular details of how that might be driven. So I will stop there and come through to a summary of what we've explored so far. So making sense of omics data can be hard. You can have thousands of data points in an experiment. And I think, at least today, we've shown you that it's relatively straightforward to upload and look at the data set through ClueCore Omics Explorer. And we've been able to, and Anne was being able to show this subset analysis through those PCA plots to show how the data can separate and which genes are driving that separation, which ones are significant. And that was important because it validated at the molecular level the same pattern that was seen in the microscopy, that P53 mutation at least is contributing to some extent in that response. Then as we move on to MetaCore, we've shown that aristolochic acid exposure is useful for identifying, using the biomarkers as to identify toxic processes, and those coincide with what we were expecting. We identified HNF4 transcription factor as a key upstream regulator of this toxic response that was in common, irrespective of P53 status. And then we've identified MDM2 as a regulator of the P53 dependent protection here. Okay, so combining these two tools helps to make an easy workflow to analyze the data and then gain biological insight into that data. And with that, thank you for your attention and open to questions.
Yeah, and please use the chat. Okay, so here is one question regarding the types of data that we support. So in this example, we showed you microarray data, but from ClueCore's side, we also support a number of different other data formats, such as RNA-seq data, single-cell RNA-seq data, metabolomics, proteomics, microRNA, qPCR, and so on, and also a combination of these data types in multi-omics data sets. And from MetaCore side?
Yeah. So what we did today was we focused on differential gene expression using transcriptomics. But yes, MetaCore will accept and allow you to analyze metabolomics, proteomics, transcriptomics, and genomics, gene variant data as well. And as you mentioned as well, that it's really important to be able to do multi-omics, where you can layer on from the same sample set where you've done different omics experiments and layer them all together and then do this type of workflow analysis. So if you can validate with genomics and metabolomics and transcriptomics the same results, then it provides a really strong evidence set. So yes, these are accepted, and this type of analysis is possible in MetaCore.
Mm-hmm. Then I think the next question is for you, Peter, so you take it.
Oh yeah, yeah. Okay, so right, the biological. Can I access ClueCore through the can I access MetaCore through the ClueCore tool? So for this, you do need a separate subscription. MetaCore and ClueCore come with separate subscriptions, and you'll see in a moment that you can contact either of us to find out more about that.
Yeah, and then I don't think I see more questions.
Okay.
So let's move on to the contact slide.
Okay. So you're very welcome to contact us, either of us here. Our details are here. And ClueCore Omics Explorer is available for free trial as well by following the link shown there. And we'll share these slides afterwards so that you've got access to this data, and you can write to us and with any questions that you have. So please do write directly, and we can share directly the slides, or the recording will be put up on the ClueCore website there.
Yes,
So,
exactly.
yeah. And so then, just to wrap up, I'd like to say thank you very much for your attention.
Thank you so much. Have a good day.
Right. Thanks. Bye now.
Okay, bye-bye.